Well, hello and welcome to everyone joining us for this X Talks webinar. Today's talk is entitled Vascular Phenotyping, a New Paradigm in Drug Development. My name is Corey Stanton, and I'll be your X Talks host for this event. Today's presentation runs for approximately 60 minutes, and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speaker throughout the presentation, and we'll try to attend to as many as possible. If you're joining us by GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And for those of you viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank CardiX, who developed the content for this presentation. CardiX is a digital health technology company that develops and markets non-invasive patient monitoring technologies for assessing cardiovascular health. Their pioneering StigmaCore vascular biosensing technology measures arterial health by collecting clinically meaningful digital vascular biomarkers using novel sensors. These biomarkers represent key indicators of cardiovascular health, including, but not limited to, central blood pressures, vascular stiffness, vascular age, and heart stress. StigmaCore has been deployed in multiple clinical trials sponsored by pharmaceutical companies and healthcare systems to measure arterial health. And now, let's take a moment to meet our featured speaker for today's presentation. Dr. Sanjeev Bhavnani is a renowned physician, digital health clinical trialist, and policy strategist with over 20 years of experience transforming healthcare innovation. He excels at integrating digital health technologies and AI and machine learning into clinical practice, leading teams in impactful trials, achieving FDA approvals. A strong advocate for DE&I, Dr. Bhavnani champions inclusive healthcare solutions and influences national policy to ensure equitable access to advanced medical innovations. And now, without any further ado, I'd like to hand the mic over to Sanjeev to kick off the presentation. Sanjeev, I'm going to send you screen control now, and the floor is yours.
Thank you, Corey. Thank you to our organizers, and thank you to all the audience members who are here today for this talk. Here. Thank you. So today's talk is entitled Vascular Phenotyping, a New Paradigm in Drug Development. I'm a consulting chief clinical officer at CardiX. A disclaimer, these devices and trials for using these devices will be discussed. So let's dive right into it, right? This concept of vascular phenotyping. What is it? Vascular phenotyping is the characterization of functional and structural properties of the vascular system, including arterial stiffness, wave reflections, and endothelial function, to identify distinct cardiovascular risk profiles and to guide precision therapy. Why do we need it in particular disease and drug development? Well, I think we can all acknowledge, for those who have done such studies or work in the field of cardiovascular disease, that traditional blood pressure measurements do not measure the pressure at the heart. Vascular dysfunction starts before pressure, and there is a lot of heterogeneity blood pressure changes, spontaneity in hypertension, or in any chronic condition. Any two individuals, for example, with a blood pressure that is largely the same, near 140/85, who have hypertension, they have different risk. And that's risk of CVD, risk of strokes, risk of heart attacks. And traditional blood pressures alone do not accurately measure the risk of events like a heart attack or stroke. So vascular phenotyping can be a driver for precision medicine and mechanistic insights that support drug development. When we look at any condition, there is disease progression. It goes from a normal state to a pre-disease state to then a disease state. And in each of these intersection points and in the progression, there is a period of reversibility, and then there's also a period of irreversibility. When we look at cardiovascular disease and actual heart function, so normal function to the detection of cardiac involvement can take a very long time. But it also depends on the severity of conditions that cause cardiovascular disease in people, the severity of diabetes, the severity of hypertension, the degree of smoking. And so this preclinical stage to the clinical stage, and then detectable cardiac involvement, the modalities in which we can measure the progression between the stages of disease is largely restricted to what we can use in healthcare diagnostics, using ECGs, using echocardiograms, using imaging, sometimes very difficult to get diagnostic testing. And then when done, there's no method to be able to serially do those in order to look at progression to irreversibility, but also in the regression to disease reversal. And with the advent now of mobile technologies and artificial intelligence, really from head to toe, if there is a physiologic measure that is of interest or that could be measured, it is being captured and analyzed. Whether smart rings to look at sleep and autonomic responses of sympathetic and parasympathetic nervous systems, smartphone ultrasounds to look at vascular function, sensors woven into clothing to look at day-to-day changes, right, in heart rates, in physiologic measures, particularly with exercise, and the list goes on and on. So we have new methods to digitally capture physiologic measures of interest and to be able to analyze those measures now using artificial intelligence and machine learning. So our agenda today in this in the topic of vascular phenotyping and drug development, we will use a motivating example of hypertensive heart disease. And we'll go over the physiologic measures of the arterial pulse wave. We'll speak about the analytical and clinical validation approaches for digital vascular biomarkers as we get into vascular phenotyping and the type of data analyses that can be done on such data sets. We'll look at some real-world examples of remote patient monitoring and a real-world example of vascular phenotyping. And we will then push forward into computational approaches for using vascular phenotyping within drug development clinical trials. So traditional blood pressure measurements, this probably requires no introduction. We're familiar with oscillatory blood pressure devices and oscillometric devices. And when you look at the blood pressure waveform, what we are measuring when using these devices is simply 2 points, the systolic peak and the diastolic trough. There's a number of different measures that are occurring during this waveform's acquisition and those measurements that we traditionally do not interpret. And so what are the implications of this? The field of hypertension, the drugs that have been developed for it are based on brachial blood pressure as a measure of hypertension inclusion into trials, but also drug effect. And here in an example, a randomized trial among many of the SPRINT trial, looking at intensive versus standard blood pressure control in individuals who undergo intensive treatment, who start at a blood pressure of 140, achieve a quicker blood pressure response and reduction to 120 and the maintenance compared to standard treatment. And this is associated with a 25% risk reduction in heart attack and strokes at 3 years with intensive treatment. All right, let's take this a step further. So what is the longer term impact of intensive blood pressure treatment beyond the clinical trial? So hypertension is nearly a lifelong condition. What happens as these people age? So in an extended follow-up as part of patient care after the SPRINT trial was concluded, a very interesting finding was observed that after the observational period was over, in people who had intensive blood pressure treatment strategies, that their systolic blood pressure rose back to where they were before enrollment, and at the same degree of standard treatment. So there was a waxing and waning effect of antihypertensive treatment after intensive monitoring, intensive treatment. And this wiped out that degree of risk reduction for heart attack and strokes throughout the course of after 3 years up to 11 years. So what can be the plausible reasons for why something works in a trial, but when translated into patient care, you don't see the same beneficial effects? Well, there are many reasons. there are many limitations to the use of blood pressure and this blood pressure hypothesis. There is variability, instability, and episodic hypertension. And as we looked at this graph, this curve, these are point measurements. Trial results are not translatable to the real world, and blood pressure may change, but the question is the underlying physiology also changing? The study subject risk is variable across hypertension. Phenotyping vascular risk may be the same way. So as we get now more deeper into cardiovascular physiology and hypertension, you look at the physiologic properties of the arterial pulse wave. And if you look at the animation here on the right, see that as blood flows forward, it also reflects back here. See the blood, the pulse wave is moving forward. Blood then ejects forward, but it also reflects backward. It's almost like 2 steps forward and 1 step back. So there's a propagated wave and a reflected wave. So when stiffness occurs, pressure and waveforms change. In a healthy artery, normal blood flow moves through a flexible artery. Think of an elastic band or a balloon that is very flexible. It expands out and contracts in. A stiff artery, and one is an example that has plaque, requires faster blood flow to move through it. It requires more velocity. And in this stiff artery, just as we saw in the animation, the pressure wave moves forward and it reflects back. The greater the stiffness forward, the more the reflected pressure wave moves backward. So a normal cardiac function, this is an example of central hemodynamic waveform that is normal, but the greater reflected pressure wave adds pressure on the heart. This peak increases, and therefore it manifests as an increased workload on the heart and can lead to cardiac dysfunction. So as we've looked at now, the blood pressure in the peripheral arm of 2 waveforms, of 2 points, and the blood pressure waveform in the central circulation at the aorta has similar properties, but takes into account now the physiologic parameters that determine pressure and workload on the heart, the forward wave and the reflected wave. Also the central pressure, that pressure which is exerted right adjacent to the heart in the aorta, is often different and classifies a different risk than peripheral blood pressure and blood pressure in the arm only. Cardiacs has created a number of different devices over the past over 20 years that can measure and capture central blood pressure and perform pulse wave analysis on those waveforms for measures of arterial stiffness, reflected pressure waves, augmented pressure waves. And these can be done at the point of care and now in ambulatory settings with a blood pressure cuff that people can use at home. Is pulse wave biomarkers for the first device available to be used, right, for patients and for people, is a blood pressure device called Connect that can measure central blood pressure, pulse augmentation pressures, augmentation indexes, reflected waves of arterial stiffness. So let's look closer at the analytical and clinical validation before we start discussing the use in patient care. In order for any measurement, a vascular biomarker to be used, it should be analytically validated and also clinically validated. And so the specific context of use for such biomarkers drives the extent of evidence needed for its validation. In analytical validation, it must establish performance and acceptance characteristics of that measurement. This can include reference ranges, analytical rigor, analytical performance characteristics. It's very similar to laboratory testing. And clinical validation. Establish that the biomarker measures and predicts that concept of interest. What is the study design? Is it clinically meaningful? What is the benefit risk assessment? When looking at vascular biomarkers, several large population-based observational trials have analytically validated these measures and then what is the progression of normal vascular aging. And the curves on the right are in different age groups from 20 to above 90. And what happens to central systolic pressure, central pulse pressure, measures of stiffness, augmentation pressure, and augmentation index as healthy people age. And so there's a differential effect on wave reflections and aortic pulse wave velocity as we get older. We don't capture those in brachial blood pressure alone. We know that brachial blood pressure increases as a different characteristic in that curve as people age, but what's happening to the underlying vascular function and vascular health? In a randomized clinical trial, now with looking towards clinical validation, can be used to understand from a clinical perspective what happens when these parameters change. And in a trial of over 2,000 individuals with uncontrolled hypertension at a brachial blood pressure of 160/90, those who were randomized to calcium channel blocker and ACE inhibitor versus a beta blocker and diuretic, two different treatment modalities to reduce pressure. While brachial blood pressure reduced in both, these are antihypertensive therapies, central blood pressure had reduced more in the calcium channel blocker and ACE inhibitor group compared to beta blocker and diuretic, and was associated with a 30% less cardiovascular events directly due to a reduction in central blood pressure. While brachial blood pressure reduced and were equivalent in both groups, central blood pressure was the driver for the beneficial effect of treatments. It leads to clinical validation of these markers, but also that phenotype that we're discussing, and how are we measuring risk. And so many of these vascular biomarkers have been validated in different disease states, from neurovascular to oncology, diabetes and metabolism, and inflammatory conditions, erectile dysfunction. And so we understand now that vascular phenotyping and vascular assessments is more than cardiovascular because it portends a risk of disease at the level of individual organs. So let's look at vascular phenotyping in hypertensive heart disease. And here in a video example of understanding plaque, atherosclerosis. Hypertension is among the leading causes of atherosclerotic plaque in the arteries of the body and directly in the coronary arteries of the heart. And here in this animation, again, looking at blood flow, that inflammatory measures, lipid profiles, hyperglycemia, insulin resistance, all contribute to the formation of atherosclerosis, the formation of lipid-laden plaque in the arteries of the body and in the arteries of the heart. And this is what classifies as coronary artery disease and what can lead to heart attack and strokes. While we look at that animation, we do have methods to be able to visualize this plaque in people. This is an example of pulse wave and its relationship to atherosclerosis plaque. The figure at the top is the degree of plaque. Right? As plaque increases, it obstructs the diameter of the coronary arteries. So how do we visualize this? We visualize it on CAT scans. And the example here, the heart sits in the middle, looks gray because it's soft, it's muscle and blood. The lungs are air, they look black. The spine and the ribs are white. They're bone, they're hard, they have calcium. And if I were to point your attention to this one line here, this is an artery of the heart, coronary artery, with no white in it. So the calcium score, plaque score is 0. And in different individuals, as plaque increases, the amount of white increases. And so we can visualize the degree and severity of plaque now using, as an example, CAT scan technologies. And as such, in such individuals, the pulse wave morphology is changing because, as we looked at previously, those parameters of blood flow velocity, of wave reflections and wave velocity are also changing in relationship to the degree of plaque in an artery. So, there have been several interesting studies that have looked at this concept of vascular phenotypes in hypertension. And this recent study had looked at vascular biomarkers using central blood pressure and stiffness measures, right, using the technologies that we have discussed. Also, reactive hyperemic indexes that can measure endothelial dysfunction, and in addition to ultrasound measuring flow-mediated dilatation, vascular function, and vessel thickness. So a comprehensive assessment of what's happening to vascular health, and can we measure it in people with early hypertension? Using machine learning and cluster analyses, these clinical trials have shown that there is a measurable heterogeneity in hypertension. And this heat map here in cluster diagram can now map the measures of central hypertension and stiffness with endothelial dysfunction across the severity of hypertension, across risk in individuals. And, for example, in women, the risk is different than in men. In people who have high nocturnal blood pressures, the risk is different than those who do not. But now looking at different phenotypes by understanding the vascular measures of arterial function. And multi-omic profiling using pulse wave analysis has emerged in several large biobanks, such as the UK Biobank, that has now begun to understand age-dependent associations of high blood pressure with cardiovascular outcomes, but also diversity and equity, the ethnicity of arterial stiffness. So vascular phenotyping has been proposed as exploratory endpoints in clinical trials, but also subgroup analyses to support regulatory submissions for therapeutic effect, because there's a measurable way to understand the severity of a cardiovascular condition of interest. And can this approach be used as a combination approach using digital biomarkers and therapy? So the goal of vascular phenotyping is to precisely measure cardiovascular issues. And this is our proposed model and what's being used clinically, right? In people who are using, for example, the Connect blood pressure monitor for home-based vascular assessments to understand, do their parameters fall into low cardiovascular risk, intermediate cardiovascular risk, or high cardiovascular risk. And this is in addition to and beyond brachial blood pressure alone. Let's look at some examples of using pressure in the real world in remote patient monitoring. I think we can all largely acknowledge these are not our patients out there, right? Digital natives. Our patients are more like this, right? They have the complexity of a condition, but they also have complexity in using new devices. So here is an example of an individual who has uncontrolled hypertension, a blood pressure of 180. And for those who are abroad in the United States, this individual might be seeing their physician once a year or maybe twice a year. So when we employ a strategy of remote patient monitoring, right, people using devices at home, capturing measurements and then relaying that to their care providers to close the treatment loop at initiation, and by having individuals engage in their hypertension monitoring, titration of medications, adjustment of lifestyle modifiers can result in a reduction in blood pressure towards its improvement. And we can also see as time goes on when the disease also goes back out of control. So optimize medications, change lifestyle as needed in order to do the same thing. Reduce blood pressure to achieve the treatment goal by employing a remote patient monitoring strategy. These remote patient monitoring can also help us understand individuals' behavior. In this example of a 68-year-old with hypertension and obesity, blood pressure has been high for a very long time, 150 to 160, but we can see a precipitous drop in blood pressure. And when asking the individual, well, what changed? They started intermittent fasting. And so a lifestyle modifier that a person took into their own hands while using a strategy of remote patient monitoring resulted in improvements in time to blood pressure control. And finally, using novel technologies, right, to identify patterns of disease, 24-hour blood pressure monitoring. It's very difficult and near impossible to measure nocturnal blood pressures when people are sleeping. But using new digital devices, using new ambulatory 24-hour blood pressure monitoring devices, there are methods to be able to capture patterns at night. And this example here of nocturnal blood pressure dipping, I'd like for the audience to think about someone's blood pressure is high during the day. It should be lower at night when one is more relaxed. And that's called a dipping pattern. It's a favorable sign that blood pressure can be changed. It can reduce. The opposite, if blood pressure does not dip at night, or in fact goes up, reverse dipping, portends a much higher risk of stroke and heart attack in that individual with hypertension. So let's bring this all together and look at a practical real-world example. A 48-year-old woman with hypertension. While she has healthy habits, she has a high-stress lifestyle. She also has high cholesterol. She takes medication for hypertension and hyperlipidemia. She wants to better understand her cardiovascular risk. At 48, her risk should be low. She should also have zero plaque. When doing an assessment on this individual, he, in fact, and I think the audience now is familiar with looking at CAT scans. This is a coronary artery of the heart. Heart sits in the middle. There are observable areas of white and plaque in this 48-year-old. She had to do a CAT scan in order to understand this. Well, when she captures central blood pressure and pulse wave analysis using blood pressure devices, that her brachial blood pressure remained moderately high, 143/67, but she measures with high central blood pressure. And she has an augmentation index, a measure of arterial stiffness that is very high. So she has peripheral and central hypertension with arterial stiffness. So her cardiovascular risk is in fact not low or intermediate. Her cardiovascular risk is in fact high and very high. So using computational approaches to vascular phenotyping, bringing things together now, the application to drug trials to identify patient clusters. Concept of patient similarity for precision phenotyping. Many individuals in part of their healthcare capture a lot of data. They have data related to their electronic health records, their physiologic data as part of cardiovascular imaging or any imaging modality. We use all that data and then construct patient similarity networks to identify subtypes of individuals and their relationship to outcomes. And so topographical data analysis and similarity data analyses aim to do exactly that. Cluster individuals with a condition of interest, hypertension, across the severity of the disease. Can we use a precision analytic approach in order to look at the progression of disease severity from mild, green on the left, through intermediate in the middle, to severe on the right? We understand, and as we saw very early on, progression of disease is a continuum, right? Can we use a phenotypic approach using healthcare data and digital data in order to understand this? So vascular phenotyping is patient identification and to understand treatment response. So using such digital vascular, biomarkers at the time of enrollment and understand their relationship to given individuals. Single measures of vascular biomarkers or clusters, as we saw in that real-world example. Brachial blood pressure plus central blood pressure plus arterial stiffness measurements. To look at clusters of individuals in hypertension for the objective of looking at which cluster has an early treatment response to an investigational therapy. Who's at risk of an adverse event? Are there markers in certain clusters of disease reversal? And then the pleiotropic effects of investigational therapies. And a highlight here, blood pressure and weight loss, very similar to what we've experienced with GLP-1 therapy. While there is a weight loss effect and that anti-hyperglycemic effect in diabetics, in hypertensive individuals, they see the same metabolic changes and a reduction in blood pressure. So can clustering analysis also advance how we design clinical trials for investigational therapies and how we are measuring the effect of that therapy on cardiovascular function, with the ultimate goal, objective of understanding understanding the beneficial effects on outcomes. So we covered a lot of ground today, and in summary, we went over the pulse wave and its measurements to central pressure and arterial stiffness. We looked at new technologies and devices that perform pulse wave analyses. We went through an understanding of the analytical and clinical validation for vascular biomarkers in hypertension. We looked at vascular phenotyping and its application in heart disease, including remote monitoring, and new computational approaches for vascular phenotyping and patient identification in trials of investigational therapy. I hope that this talk was educational and informative. My contact is here should anybody like to reach out or have any questions.
Excellent. Thank you so much, Sanjeev, for that very insightful presentation. We do have time now for our Q&A session. And as we go through this session, I invite the audience to continue sending questions or comments. Just a reminder, if you're attending via GoToWebinar, please use the questions chat box. And for those of you watching via LinkedIn Live events, you can feel free to post your questions using the comments tab that's there on LinkedIn. We have already received some audience questions. I'm going to go ahead and kick off with the first one, and we'll see how many we have time to get through. But as Sanjeev mentioned, there will be an opportunity for follow-up after the webinar, and I'll share the contact info again with you all shortly. So this first question that's come in from the audience here, this audience member is asking about how can digital vascular biomarkers be validated as surrogate endpoints in clinical trials, especially in the context of regulatory acceptance for drug approval?
Thank you, Corey, and thank you for the audience member for the thoughtful question. Vascular biomarkers, just central blood pressure and arterial stiffness, have been used in safety assessments for investigational therapies as part of using them as surrogate measures, right? Either, I would say, surrogate or as secondary or even primary measures as part of regulatory trials will require a thoughtful discussion with the regulatory body. These measurements have been validated among a variety of different clinical trials. Using them as tools and endpoint assessments in individual trials will require an agreement with the regulatory body for that to occur. There are activities underway to validate these digital vascular biomarkers as tools to be used in investigational therapies and in regulatory trials, and that hopefully is on the horizon in the near future.
Fantastic. Excellent. Thank you. I think we have time for a couple more questions here. So jump to the next one that's come in from the audience. And this audience member is asking about the practical considerations for integrating vascular phenotyping tools into multi-site or even into global clinical trials, particularly when it comes to standardization, scalability. Where do you fall on that?
Yes, right. Very good question, right? Right to the operations, right, of using measurements as part of clinical trials. Very good data coordinating approaches, right? When looking at whether it's digital vascular biomarkers or imaging, EKG, electronic health record, these are different formats. These are different ways of collecting the data. Also, the serial measurements, right, over time in given study subjects during the course of the clinical trial will require first and upfront a robust data coordinating plan, statistical analysis plan, and then to use interim analyses to understand if there's any missing data, understand, right, if there's anything that needs to be done differently when capturing high frequency measurements over the course of the clinical trial. So these measurements and vascular biomarkers have been used as point assessments over time in people in trials of pre-post therapy, before and after an investigational drug, but will require that the data coordinating center be able to capture those appropriately in individuals and then also analyze them. On the analysis front, there are different ways to be able to now look at repeat measurements in single individuals in trials, but also in clusters. And that can be by employing hierarchical clustering, the example that I showed of the Manhattan plot, using the current neural networks, using different approaches, even deep learning, to be able to understand patterns in using vascular biomarkers and underlying risk and underlying treatment responses. So a very good question. Leads right to the operations of using multiple and new measurements as part of data coordination in trials.
Fantastic. Thank you for your really detailed response there, Sanjeev. I think we've got time to hit one more question here before we wrap up. And this audience member is asking, perhaps you could just provide some examples of how vascular phenotyping has influenced, a go no go decision in early phase trials, especially with in cardiometabolic indications.
I think that's also a very good question, right? There is a lot of interest in using these measurements, right? Particularly in investigational therapies and early investigational therapies to look at signal, right? Of where are there different patterns, right? Of disease or response rates to an early phase investigational therapy. And so as far as go or no go, I'd probably rephrase that into how is it being employed in order to improve the efficiency of that clinical trial? Whether it be early stage, phase 1, phase 2 type of clinical trials, but looking at the objective of using these measurements in that particular phase of clinical trial to understand signal and be one, to understand risk, side effect, or safety in another. And so just as the field of cardiovascular, even oncology is led here by understanding the phenotypic clusters of cancer, of cancer genome sequencing, of inflammation, using multiple measurements. But in a similar way, look at digital vascular biomarkers as a clustering approach to understand safety, adverse events, and also drug response rates. I think is a way to approach this as far as using it in clinical trials.
Excellent. Thank you so much again for a great detailed response. Unfortunately, I think that is all the time we have for our Q&A here today. But if we couldn't attend to your questions, or if you have any further questions, you can, of course, get in touch with Sanjeev using the email address currently there up on screen. That's his email there at Cardiacs. I have a chance to follow up with you after the webinar. Same goes for anyone who's watching the recorded version of this webinar. Feel free to reach out if you have any questions that you'd like responded to. I do thank everyone once again for participating today. If you're attending by GoToWebinar, I've added a link in the chat box where you'll be able to view the recording of this event, and you can, of course, share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from XTalks with access to the recording, and after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated, as it helps us to improve our webinars. For those of you watching on LinkedIn Live, just a quick note, we do encourage you to create an account on xtalks.com and register for this event, which will not only give you access to today's recording, but to our entire library of content, which includes another great Cardiacs webinar that we do with Sanjeev, I believe it was last year. So you can register and you'll be able to access that as well. Before we sign off, I just wanted to thank you, Sanjeev, once more for your excellent presentation. Thank you for dealing with the elements today. Really appreciate it. We hope you all found this insightful and informative, and we wish you all a great rest of your day. Take care, everyone. Thank you.
Thank you, everyone.